Good afternoon to all the ladies and gentlemen, esteemed colleagues and honored guests to the post-lunch second plenary session. I, Ms. Nikita Dhaigude from B Farm first year, feels immensely privileged and honored to introduce our esteemed guest, Mr. Dattatreya sir. Welcome, sir. Sir has completed his D.Pharm in 2005 from North Maharashtra University. Then, Sir has completed his B Pharmacy in 2008 from Dr. Babasaheb Ambedkar, Marathwada University. He went on to earn his MS in Pharmaceutical Analysis from the National Institute of Pharmaceutical Education and Research between 2008 and 2010. Sir achieved a significant milestone by qualifying GATE in 2008 with an impressive score of 99.39. Furthering his academic pursuits, Sir is pursuing PhD from Dr. Babasaheb Ambedkar Marathwada University, Chhatrapati Sambhajinagar. Additionally, he was awarded the prestigious Sri Ratan Tata Trust Scholarship during his third year of B.Pharm. Sir also successfully completed the basic course of intellectual property rights conducted by WIPO in 2009. Mr. Vani, who is our alumni, Mr. Vani embarked on his illustrious career as a research project trainee at the prestigious Indian Institute of Chemical Technology in Hyderabad during the years 2009 and 2010. His foundational experiences there paved the way for a notable 6-year tenure at Wockhardt Limited, where he progressively advanced from research associate to senior research associate and ultimately to the role of research scientist. There, he has awarded as a Rockstar Award 2 times. Following his impactful contributions at Wockhardt, Mr. Vani joined Mylan Laboratories Limited for 3 years, where he distinguished himself as a team leader and senior executive. In Mylan, he worked at the Global Injectable R&D facility in Bangalore. He was awarded as a business performer for 2 consecutive years. He continued to demonstrate his leadership abilities as a team leader at Viatris from 2020 to 2021. His exemplary leadership and expertise led to his current role at Endo International PLC. Over the past 4 years, Mr. Vani has demonstrated exceptional dedication and skill, earning a promotion from deputy manager to manager. With a robust background in research and development and a proven track record of success in leading scientific teams, Mr. Vani continues to be a beacon of excellence and innovation in the pharmaceutical industry. We are privileged to have him with us today, and we look forward to the insights he will undoubtedly share from his extensive and varied experience. Advanced analytical characterization techniques encompass a diverse array of methods used to study materials at the molecular and atomic levels, from spectroscopy to microscopy and beyond. These techniques offer profound insights into the properties and structures of substances, aiding various fields including chemistry, material science, and biotechnology. We invite Mr. Vani to share his insights and expertise on this fascinating topic. With this brief overview of this topic, advanced analytical characterization technique, I kindly request Mr. Vani to continue this session. Thank you, sir.
Thank you, Renika, for this kind gesture and introduction. So, first of all, I will be thankful to the principal Angadi sir and the committee of the Asian Institute of Pharmacy for giving me this opportunity to deliver some talk on this advanced analytical calculation techniques. Hello. Yeah, I'm audible to all. Hello.
Yes, sir. You are
Yeah, I'm
audible.
audible.
Yes, sir. You are audible. Your presentation is also visible.
Okay, can I continue?
Sure, sir.
Yeah, okay. Thank you. Good afternoon, all. Yeah, this would be the quite difficult session for me because everyone have completed their lunch. So bear me for that one. So I am going to continue my session. So here I am covering this overview of advanced analytical characterization techniques. So basically, I'm taking the overview means I will generalize all the techniques and their importance in the pharmaceutical analysis. So the topic I will be covering here, why it is required to characterize the pharmaceutical molecule or its molecule, that I am giving to the briefing. Then what are the techniques we are going to use for the characterization, mainly spectroscopic, thermal, then some advanced hyphenated techniques that I am going to enlist. Then mainly I'm focusing on the peptide characterization. There is a current demand, why it is demanding the peptide characterization and biopharmaceuticals. So I'm going through the structural characterization of these peptides, mainly primary, secondary, and tertiary. Along with that, the major problem that will focus on this peptide and protein is related to the aggregation. That going to I will cover, and what is the regulatory requirement for characterization that I'm going to cover. So first slide, I'm going to the requirement of analytical techniques, where it is required. The analytical technique is required basically from your drug discovery to all the stages of your API as well as the pharmaceutical industry. In drug discovery, where the analytical technique is required. The analytical technique is required mainly to analyze the raw material as well as the in-process sample, or to identify the potential NC candidate, means new chemical entity or new molecular entity. Because when we are going to the discovery, there are hundred of molecules we have. To scrutinize that molecule to the lead molecule, we have to require the robust analytical techniques. So by using this analytical technique, we are going to optimize the molecule and we will get the lead candidate, and from the lead candidate, we will go for the approval. And where it is required, it is required in the bioanalysis. When we are in the discovery, you should do the bioanalysis of your molecule by using the bioanalytical techniques, where you are using LCMS as the major technique. Then it is required in the PKPD analysis, where you can inject your molecule as a lead molecule in mice or rat, and you can analyze this sample and get this PKPD data or adsorption data. It is also requiring toxicological study, where you finalize your pre-formulation. You have to inject your pre-formulation in the animals, and you can get this toxicological data by using a different analytical techniques. So this is not only limited to the discovery. You require analytical techniques in the drug development also, where it is required in the drug development. Generally, in the pharma industry, drug development, it is related to the API development as well as this formulation development. So in the both the field, you required your analytical techniques. In the API, you required analytical techniques for analysis of your API as well as the intermediates, as well as their in-process monitoring. You require the robust analytical methods, analytical techniques. So in the formulation, you should have oral as well as the parenteral complex formulation. In every stage, you require analytical technique. By default, you can stay as the analytical technique is nothing but the heart of the pharmaceutical industry. Without that, you can't survive. You can't develop any new molecule or new formulation. there is some regulatory requirements where we require analytical techniques or analytical characterization. In the pharma, we are majorly working on this IND submissions, NDA submissions, or NDA submissions. When you are doing the IND at the stages from initial to the filing, at each stage, you require different analytical techniques and you have to submit that data to the regulatory. In the NDA is nothing but the branded drug. When you are working with the branded drug, you require each stage of submission or each stage of our development, you require these techniques to qualify the formulation or to optimize this formulation. Same with the generic. In the generic, nothing but generic is not but the copying of our NDA, means branded drug molecules. When you are developing any NDA molecule, at that time you also supposed to require or characterize our molecule with the NDA. NDA is nothing but the branded drug. It's in our line pharma we call as a reference listed drug or reference standard. We have to compare our generic product with the reference listed drug. It is nothing but the Q1 Q2 similarity of our NDA with NDA. So to prove that Q1 Q2 at each stage, at different stages, you should compare our NDA data with the NDA and you have to submit this data to the regulatory agency. In similar way, 5052 or NC submission, we require these analytical techniques for this submission. So these are the different spectroscopic characterization technique that generally uses in this pharmaceutical field. UV visible spectroscopy, infrared spectroscopy, near infrared spectroscopy, Raman, NMR, X-ray, atomic absorption, atomic emission, CD, circular diagonal spectroscopy. In that, you have to work on the different region, far EV or near EV. Then fluorescence spectroscopy. These are related to spectroscopic characterization technique. As well as there are some thermal characterization techniques that we are using for the characterization of a molecule. In thermal characterization, your material physical and chemical properties are changes with respect to the temperature. So in thermal characterization, we are going to use thermogravimetric analysis, differential thermal analysis, differential scanning colorimetry, thermomechanical analysis, thermo-optical analysis, direct relaxation spectroscopy. These are the nothing but different thermal analytical technique. As well as that, some of the hyphenated techniques that we are focusing on characterization molecule is the ICP-MS, LC-MS, GC-MS, LC-MS-MS, GC-MS, SAME, TAME. These are the techniques that we are using to characterize our molecule or to show the sameness of our generic molecule with the innovator molecule. So the next part will be I'm focusing on the peptide characterization. What is peptide? Peptide is nothing but a polymer which having the about 14 amino acid, means less than 40 amino acid that are connected to each other through the peptide bond. And so, and this different peptide with connected to the peptide bond and give to the proteins. So in the pharma, there is a major challenges to characterize the protein peptides. So this is the amino acids. Generally, we have the 21 amino acids. There are some are essential and some are non-essential amino acids. So in the peptide, what we have to focus? We have to focus on the peptide bond. What is the peptide bond? Peptide bond is nothing but the linkage of the carbonyl group of this one amino acid and the amino group of the second amino acid. These two groups will link together and they will form the peptide bonds. Depending on the linkage of the amino acid, there are different types of the peptide. Dipeptide, tripeptide, tetrapeptide, oligopeptides. If it will contain more than the 10 amino acid, that will be the oligopeptide. In the pharma industry, you will majorly focus on the oligopeptides because you have the molecule having more than 9, 10 or 29, 30 amino acids. Then these are the different glycopeptides and lipopeptides. When sugar moiety or lipid moiety is connected to your peptide or amino acid sequence, that will give the your lipopeptide or glycopeptides. So generally, you have to characterize this all your lipopeptide, glycopeptide, or what were the peptides you are getting in this one. So this is the example of the tetrapeptide. Here, 4 amino acids are linked together, and this is forming a tetrapeptides. So why it is needed to characterize the peptides? So generally, peptides are formed by two techniques. as well. First one is the recombinant, and second one is the synthetic. If some innovator has filed his product by using the recombinant DNA technology, and the generic developer want to use the synthetic peptide and launch their product, so they have to prove this sameness of their synthetic peptide with this rDNA peptides. So generally, we are getting mostly the synthetic peptide. So why peptide injectable formulations? Why it is needed to peptide only in the injectable form? Why not in the oral formulation? Because if you are taking peptides are highly labile for the degradation. When you are taking the orally, it will get degraded by using the what were the different types of the enzyme in our intestine or stomach that will degrade that molecule, and it will not show any activity or any potency. So for that purpose, generally the peptide formulation are in the injectable form, not in the oral formulations. And second, why it is in the injectable and why it is needed characterized, because they have the high potency. If you the dose, if you are seeing the peptide, it is in the nanogram or milligram range. So to characterize that accurately, we require analytical technique. They have the selective activity, they have the broad range of target, low accumulation tissue, and they have the high chemical and biological diversity. For this purpose, we require the peptide-based formulation in the injectable. That may be the liquid injectable, lyophilized injectable, or some novel drug formulation by using different types of the polymers. So the first peptide that you all are discovered in the industry is nothing but the insulin. Insulin is the peptide having the 51 amino acids that are linked together by different disulfide linkage. This is the requirement of peptide characterization. Why peptide is characterized? Peptide is nothing but the long chain of the amino acid. So it is considered as the polymer or is it biopharmaceuticals. In the peptide, there are when the same peptide is the linkage of the same amino acids, then it will be the homogeneous sequences. Or when there are the different sequence of the amino acid are interlinked with each other, that will give the heterogeneous sequence. So to prove that our peptide is homogeneous or heterogeneous sequence, we require the characterizations. And third is the peptide has a complex shape because they have the folding and defolding requirements are there. And one more requirement is the regulatory requirement. When you are developing any ANDA, you have for the generic sponsor should provide this sameness data with your R&D. R&D sameness data is for both. In the previous slide, I have mentioned API. API is nothing but in this in the generic development, we are using synthetic API, or the NDA is using the recombinant API. In that case, we have to prove the sameness. It in the way we call it as the biosimilarity of our API with your RLD, innovator drug formulation. In that, what we are characterizing, we are characterizing the primary or sequence of physicochemical properties, secondary structure of our molecule, tertiary structure of our molecule, and the peptide have the tendency to get oligomer aggregate formation. So we have to study that well. And one is the biological activity. We have to prove with the bioassay or bioequivalency. And one more challenges with this is the impurities. So there are regulatory guidelines are there. In that, we have to impurity profile of our ANDA and RLD should be the similar. If you are getting any impurity in your generic product more than the innovator, at that time you have to characterize that impurity, you have to qualify that impurity. So that will be tedious. So generally, during the formulation development, we have to target such that our impurity profile should be comparable or less than the innovator product. So this data we require during the submission. So what peptide structures? Peptides generally were characterized in the three forms. There is the primary structure. Primary structure is nothing but the sequencing of your amino acid, how the each amino acid are connected to the each other. If amino acid sequence is nothing but there is a linear sequence. If innovator have the 10 amino acid in the same linear manner, our API end API should have that amino acid sequences. If there is any interchange of this amino acid, that will lead to the different activity or different profiling, impurity profiling. Then you have to study the secondary structure. Secondary structure is nothing but the folding and defolded of our amino acid, because amino acids are the complex structure. They will interact with each other and they form this alpha helical, beta C, turn, or random structure, random coil structure. Tertiary structure. Tertiary structure is nothing but the link amino alkyl group of the one amino acid is linked to the different amino acid, and they will fold it with each other and they will give the tertiary structure. So we have to characterize all these primary, secondary, and tertiary structure in our molecule. So these are the enlisting some of these structural characterization techniques. In primary structural characterization, we are using capillary isoelectrical focusing, intact mass, then peptide mass fingerprinting, amino acid mole ratio, capillary electrophoresis. For the secondary structure characterization, we require FTIR, far EVCD, Raman spectroscopy, EV spectroscopy, near infrared spectroscopy, Raman spectroscopy, UV, and mass. These are the secondary structure. In tertiary structure, we are using the fluorescence spectroscopy, near EV, 2D NMR, X-ray crystallography, and molecular modeling. So now we are going with the one by one techniques. So capillary isoelectrical focusing. Capillary isoelectrical focusing is nothing but to determine the primary sequence or primary characteristic of our molecule. In this way, in this we are determining the isoelectrical point. What happened in capillary isoelectrical focusing? By giving the different pH gradient, your molecule get interacted in this capillary, and they will force to drive in this capillary, and they will retain at a certain retention time, and that will, based on that retention time, you can calculate the pi value or isoelectrical point of our molecule. That can be detected by using the UV detection techniques. So in this development, first you have to run our marker. Marker is nothing but they have the fixed pi values. If you are getting the fixed pi values, if you are, you can consider that your instrument is okay, then you have to inject your sample. So sample is prepared in by using the different buffers or stabilizers, and you have to inject that one. So based on this, we will get the isoelectrical point. So your isoelectrical point should match with your theoretical value or with the reference product isoelectrical point. So based on this, you can confirm that our APIs or product is primary characterized. Intact mass. Mass is nothing but LCMS. LCMS is nothing but LC is connected with the mass, high resolution mass spectrometry. It is nothing but the law between the fish and parrot. Fish is nothing but our HPG system, that is in the liquid phase, and our mass is in the solid air phase, means gases. So our liquid will get transferred into the mass spectrometry. In this link, this HPLC is linked with the mass by different ionization source. That is the ESI, electron spray ionization, chemical ionization, APCI, MALDI. These are nothing but ionization source, where your liquid get converted into different droplets. That droplet get carried to the mass spec, and in the mass spec you will get a different masses, means monoisotopic mass, base peak, parent peak. So in the peptide and protein case, you have to quantify the monoisotopic mass. And monoisotopic mass is nothing but the actual molecular weight of our compound. If our peptide or proteins have the long molecular weights, so in that case, in the mass spectra, we'll get a different charge spectra. Charge spectra is nothing but we will get, suppose our molecule is a 4,000 Dalton, so you will get some 2,000, 1,000 Dalton. In that, you will get the charge state, means doubly charge, triply charge, or quarterly charge spectra. So this is the one of the spectra that we will get in the charge form. The third technique is the peptide mass fingerprinting. Peptide mass fingerprinting is nothing but a confirmatory or identification test of our peptides or proteins. In this, you will get the amino acid, which amino acid are present in your peptide or proteins, and their ratio, means percentage, at what percentage they are present. Here, what we are doing, we have to digest your sample by using the different types of the enzyme, trypsin, lysine. You will digest at 37 degrees centigrade, and you have to optimize the time and temperature. And then based on this development, we have to arrest this reaction by adding some formic acid or something, because reaction will take 2 to 3 hours. After that, you have to arrest this reaction, and you will get the digested peak, means different types of the amino acid in your spectra, or different types of the fragments. So with this technique, what we can get, if you are running with generic or reference product, so you will get the overlay of both this spectra. This is the one spectra I have taken. Here is the overlay of our generic and parental NDA formulations. So here what will happen, if in your product any impurities or any deformations are there, that you will get through the different impurities or the peaks. Generally in the protein, what will happen, there is the deamidation, means some of the amino acid will hydrolyze and give the deamidate impurity, or it is oxidized, as methionine or cysteine are highly prone for the oxidation. So oxidative impurity will get. So based on this, you can get some idea our deformation of our peptides. Now we will go for the secondary structural characterization. So secondary the FTR is the basic technique we are going to use for the secondary structure of our peptides and proteins. So, generally, FTR is used for the both solid state or liquid state analysis. So, generally, in process sampling, also we can go for the FTR analysis. It will give the idea of our freeze dried or lyophilized product. So, based on that, you will get at the different frequencies. Based on this frequency, you can characterize your molecule. So there are some limitations of this FTIR. It is a semi-quantitative technique. It is not a quantitative, will not quantitate exactly the number of impurities or not. It should have the low resolution. And sometimes the protein have the ability to aggregate. So you can't determine the aggregation by this FTIR. So only secondary structure we can go with the FTIR techniques. And third one is the when you are applying the FTR is two concept. One is the KBR and one is the ATR. When you are going the KBR pellet technique, during the pelletization, there is the possibility that your protein or peptide molecule will break and it will lead to the structural deterioration or aggregation. So for that purpose, we are going for the ATR techniques. This is generally in the peptide formulation for the protein and peptide. Generally, we are getting the 9 vibrational frequencies. Based on these vibrational frequencies, we can assign some numbers to this molecule, and we can conclude that which seeds we are getting. For a beta seed or beta turn, we are getting the molecule at 1635 nanometer frequency. For random coil, 1651 or 1539 nanometer frequency. These are the in the literature there are some 8 characteristic frequency. So based on that, we can match or we can conclude this somewhat secondary structure of our molecule. In some cases, we have to derivatize our spectrum. You have to take the second derivative. Second derivative will give the some exact idea. Here in what happens is some of the application where we can our protein formulation is generally formulated with different type of the sucrose or sugar moiety. So we can study the excipient and API study by using this technique, and we can scrutinize the which sugar is suitable for our molecule. Near infrared spectroscopy. This is some of the robust technique than FTIR. We can give the secondary structure of our protein and peptides. Generally, this technique is used for the amorphous formulation where our molecule is amorphous, because it didn't show any moisture absorption. If you are considering the FTR, you are getting the water peak or moisture absorption peak, so that we can nullify in the near infrared spectroscopy. Near infrared spectroscopy advantages of FTR is we can use in the reflectors mode. FTR we can using only transmitters mode. So directly you can analyze your lyophilized vials. So you can utilize that sample for the different analysis techniques. So sample disturbance would not be there. So this technique, we can use this secondary structure and crystallinity of the sample, as well as life cycle optimization of the water content. We can determine by near infrared spectroscopy. So we can use here as a residual solvent analysis. We can take there. This is the some of the secondary derivative spectra. Here, the two studies are there. These are the different concentration of the sucrose. So, impact of the different concentration of the sucrose we can study with our molecule. or because protein have the temperature dependent degradation. So this is the temperature study at 25 degree or 80 degree. What will happen to our molecule? That is representing in the second derivative spectra. Raman spectroscopy is also used for the secondary structural characterization. Here also similarly, where it is based on the vibrational reason. In this study, its advantage is that we can get some type of the idea about the aggregation of this molecule. So it is based on the principle of the molecular polarizability. There is a change in the polarizable index of a molecule. Based on that, we can get the different frequencies and we can predict or can conclude the structure of our molecule. This is the some of the literature based reasons of the amides for the Raman spectroscopy. This is the Raman spectra that we are getting. This is nothing but native means our molecule with some of the molecule is denaturated and one with some other excipient. So when you are getting this high 195 region, then we can confirm that there is some alpha helical structure of our molecule. When you are getting the amide 3 bond, it will represent some beta seed disorder spectra. So based on this, we can conclude some secondary structure of our molecule. This is the EV spectroscopy. This is the some common and feasible technique. Generally, when you are taking the EV spectra, it will not give a good idea of our molecular or this. But when you are going with the second derivative spectra, derivative spectra of the aromatic residual amino acid will show some sharp peak in this 284 and 294 region. So in this, we can study that different pH or different ionic concentration and we'll give this spectra. So this spectra is mainly because the aromatic amino acid, mainly phenylalanine, tyrosine, and tryptophans. This is the one of the most important technique for the peptide characterization is nothing but a circular dichromism. Circular dichromism technique is based on this chirality of the molecule and optical activity. In this, what will happen? We will pass our linear light to this photo. There is a one model in this instrument is a photoelastic modulator. What photoelastic modulator will do? Photoelastic modulator will convert your linearly polarized light to the circularly polarized light. Because linear polarized light will not give any spectra or any observation in this result of this. So we have to convert linear light into circularly polarized light. For that purpose, we are using this photoelastical model. So it is not, this technique is based on this absorption of this left and right circularly polarized. Here I have given this one. So it is based on same principle as our EV spectrophotometry. It is Lambert's BS law. The absorbance is directly proportional to the molar absorptivity, path length, and chromophoric concentration. So here, during optimization, you have to play on the path length of our covate or concentration of our molecule. Because some molecules are highly concentrated, so you have to reduce the concentration. So based on that, you will get some absorbance. Here, this is the ellipticity angle that I have told in the previous slide. So in circular polarized light, you will get some ellipticity angle. It will depend on the left and right rotation of our molecule. When it is equal, then it will nullify. It will not show any ellipticity angle. So this is circular polarized light will get in the different directions and will give some ellipticity angle. So we have to calculate that ellipticity angle and we have to measure that one. So where it is absorbing? Already we have mentioned that peptides are formed by the linkage of the different amino acids and they will form the peptide bonds. So there are the two main transition in this CD spectrophotometry. That is the pi-pi transition. It is occurred at 190 nanometer. So you will get the sharp peak at 190 nanometer. And N to pi transition, that is not a stable or unstable transition, we can call it at. And you will get some negative spectra at 210 or 220 nanometers. So based on this, we can confirm our random coil or beta seed molecule. So it will give idea of our alpha helical beta seed turn on unordered structure of the molecule. So which factor you should consider during circular diagram method development? First of all, the solvent effect. Which solvent you are going to use for solubilizing of our molecule or sample diluent? So that solvent should, our molecule should be soluble. That solvent should be transparent in the range, whatever the range you are working. It should be optically inactive. If it is optically active, then itself you will show some absorbance and that will mislead you the result. So it should be optically inactive and it should have the sample solubility. Whatever the peptide or proteins we are going to use, it should be solubilized. So here generally we are using the different buffers to optimize as a solvent. Second to optimize is the digital integration time or scanning speed. You have to keep the different scanning speed or integration time to optimize your spectra to get the better S by N ratio, as well as accumulation. You have to so our signal is directly proportional to our digital integration time and accumulations. And unit, whatever the unit you are reporting. So generally ellipticity angle we are measuring in CD spectrophotometer. So application of this CD spectrophotometer. So CD is generally the confirmatory technique for secondary and tertiary structural characterization. So as I already mentioned, the pi-pi bond interaction will show you the 190 spectra. If it is showing the 190 spectra, so you can conclude that our secondary structure is the alpha helical structure is more in our molecule. If we are getting this the negative spectra, that will give the conclude that it is in the random coil conformation. So you will get the secondary structure confirmatory with this CD spectrophotometer. In some cases you don't have the chiral chromophore in your molecule. So generally during formulation you can add some chromophoric peptide in your molecule to characterize our molecule. This is the near EV. Near EV will give you the idea of the tertiary structure. It is based on nothing of the aromatic amino acid. Tryptophan will give the signal at 290 nanometer, tyrosine will give at 275 to 2 nanometer, and phenylalanine at 255 to 2. It will give the tertiary structural confirmation. So if our molecule, from this technique, we can conclude that our peptide is in proper configuration. If there is any alteration in the secondary and tertiary structure, we can get the idea about the denaturation or degradation of our peptide by the CD spectrophotometer technique. Fluorescence spectroscopy. It is used for the characterization of the tertiary structure of molecule. Here what will happen, the molecule get excited at 380 nanometer and give the emission spectra of the 350 nanometer. So it is same principle whatever we are using in the fluorescence. First it get excited, then excited state, and it give the fluorescence emission spectra of our molecule. So this is generally some application. In the if you are seeing the diagram A, here what happened, this is the native state of the molecule when you are manufacturing our batch, and we have kept a sample at different time interval, 1 month, 2 month, 3 month, at 60 degrees centigrade. If you are seeing the intensity of our main peak structure, that is going to the going is decreasing as the time is passing, the intensity is decreasing. So it is showing that there is a deformation or denaturation of the peptide at 60 degrees centigrade. This is the diagrammatic representation of your absolute values. This is same with comparison when we have the two product at different ages. So from this you can compare the age of our sample as well as generic drug. We can compare with the innovator formulation. If this is a run of, you can consider innovator or generic, so it should be similar intensity as well as this 350 nanometer emission. If intensity is dropping, means you can conclude that red is have the high potency than the green signal, because green have the low intensity spectra at 350
Hello, no
nanometer.
sound. Hello, no sound.
Hello. Yeah, it's audible.
Yes, sir.
Yes, sir, audible.
Yeah, someone is told that no sound. Okay, thank you. Yeah. Okay. Okay, it's going well, na? Not fast or slow.
No, sir. Well,
Yeah, thank you. So this is the emission spectra. From this, we can get idea of our molecule. Here also you can see the some deformation is there in this product. So fluorescence spectroscopy will give you the tertiary structure of this peptide and protein, and it will require for the long-term stability condition. Whatever the spectra I have presented, based on this, if your spectra is similar at different time interval, means at that time your molecule is stable. If there is some deformation or loss of the intensity, from that you can predict that there is some deformation, denaturation happen to your molecule. This is the one technique that is used for the secondary tertiary characterization, solid state hydrogen deuterated exchange. Here what happened? There is a you have to exchange our hydrogen with deuterium. So in this technique, you can use a desiccator or some instrument. In the control humidity and temperature, you can exchange our hydrogen molecule with the deuterated, and the deuterated molecule you have to pass in the mass spectrometer. So you will get the deuterated mass spectra, that is the different M by Z or different charge species. So based on this, you can conclude or you can calculate some structural species of a molecule. So these are all the spectroscopic technique that we are using day to day for this structural characterization. So now we are going with the thermal analysis. So basic principle of the thermal analysis. Hello? Yeah, anyone is speaking? Hello? So thermal techniques mainly consist of our
Rishi sir, are you participant?
can I continue or where to stop? Hello? So in the thermal analysis, we have the sample holder or sample compartment. Sample compartment is sensor. In the thermal, we are using the thermocouple as a sensor. One is the temperature control furnace we have to use that will give the computer to collect this data. This is the basic diagram of each thermal technique, whatever we are going to use, that is the DSC, DTA, PG. So first one is the thermogrametric analysis. What will measure in the TG? Thermometer with it will measure the change in the weight with respect to time and temperature. So in the thermogrammetric, which parameter we are using? We are using the thermobalance. Then the spectra or the data we are generating with this TJ, it is known as the thermogram. In this previous slide, I have mentioned there is the furnace is there. Sensor is nothing. Thermocouple is our sensor. So mainly it is depend on the heating rate, at which rate you are heating your sample. So based on the heating rate, you can get our thermogram. So which factor you should consider? One factor is the purge gas, because if you are using you have to use the inert gas as the purge gas. If you are not passing any inert gas, so the air or air what is there, that will get oxidize your sample and that will give the mislead thermogram. Or you have to state the calibration state of our instrument to get the proper result. In now scenario is our TGA thermogram will connected with some hybrid technique like some of the spectroscopy like IR or chromatography like GC or AMS. So when you have to pass our gases or vapor from TGA to this second technique, so you will get some characteristic band or characteristic frequency. So that will give you some conclusive idea that will help in your structural characterization. This is one of the most important technique that we are using for this characterization is DSC. In DSC, what we are measuring? We are measuring the amount of heat required to increase the temperature of sample and reference. Here you have to measure the heat for the sample as well as the temperature, and you have to calculate the differences of our difference of samples. So in the DSC, main is part is our cell, where we are keeping our sample. That is the reference and sample holder. And then the cell is connected with the gas inlet. As I have explained, we have to pass the purge gas. Based on this cell, there are the different types of DSC. That is the heat flux DSC. Here, sample and reference are get separated. Power compensation means we are using the same cell or capillary holder for this sample as well as the reference. And nano DSC, that is the we are using for this protein and peptides analysis. In DSC, what we can get? In DSC, mostly you can get the endothermic or exothermic peak. From this, we can conclude that if there is a protein denaturation, it will give the endothermic peak, or aggregation, you will get the exothermic peak. So both aggregation as well as the protein secondary tertiary denaturation, we can conclude from the DSC data. So DSC thermogram mainly consists of our glass transition temperature, glass transition size, crystallization temperature, crystallinity, and polymorphic transition. This technique is mainly used for the polymers. So this is generally a DSC thermogram, how we are getting. This is our first page is the glass transition temperature. Then you will get some crystallization. If our molecule is crystalline, then you will get some of the melting related thermogram. If your molecule is amorphous, you will not get this melting related peak here, endothermic peak. So this is the exothermic or endothermic peak. Like this thermogram, you will get in your DSC. What is glass transition temperature? Glass transition is due to the presence of some amorphous structure in your sample. Why it will happen? It will give because of the some heat change, or there is a change in your sample because of the heating rate. So amorphous material, as I explained, amorphous material do not show any crystallization peak here. So you will get some glass transition state only in the amorphous spectra, amorphous material. So this is related to our characterization of the second tertiary characterization. The one of the most challenges is the aggregation that generally happen in peptide and protein formulation. So this is the regulatory requirement. You have to show the aggregates in your product. So you have to prove that there is no aggregation in my product up to the shelf life of this product. So agency will not accept this aggregation technique with only single method. So here we have to use some orthogonal methods. Means we have to prove with other methods, complementary method. So we have to submit both the method data to the agency on the aggregation studies. So one of the most important is the SECMALS. SECMAL is nothing but a size excision chromatography with multi-angle light scattering detector. That is the most common use technique for the determination of the aggregates to our molecule. The one disadvantage of this SECMALS technique is that when you are using the size in the column, there is a possibility to bind our molecule to this column, and that will mislead the result. As well as when you are diluting the sample and you are loading on the column, because of this dilution, there is a possibility to dissolve that soluble aggregates, and you will get the improper data. So to conclude, to prove this, you have to use the complementary or orthogonal technique. That is, we have the SVAUC, sedimentation velocity AUC samples. It is based on the sediment. Here, what will get? You will get the sedimentation coefficient of our molecule, or you will get the monomers or multimer. Monomers and multimers, nothing but the dimer, tetramer, pentamer, or 100 mers of our molecule. Means aggregates. Longer the aggregates, any aggregates we can capture in this SVOC technique. Though these are the factors on which peptide aggregation is depend on the peptide concentration. suppose your molecule is highly concentrated, so there is a chances of the more aggregation. So for that, if it is dilute in the diluted state, so there is a less chances. Yes, amino acid sequencing, pH and net charge. Change in the pH or net charge, you will get this aggregates. There is some chemical degradation or chemical modification leads to the formation of the aggregates. Different types of excipients that will also lead to the formation of the aggregates. During your manufacturing, when you are applying different temperature, pressure, or agitation and life cycle, that will also leads to the formation of the aggregates. These are the some are the internal factors that leads to the conformation, how these aggregates will form. When these factors will applied in your molecule, there is a change in the conformation. Conformation changes mean nothing but there is change in the secondary and tertiary structure of our molecule. The change conformation will lead to the formation of this nuclei, and this nuclei will form the aggregates. These aggregates will further link with each other, and they give the more big aggregates, or we call it as a fibrils. These are some of the aggregation characterization techniques that we are using for the characterization of the aggregates. ACEV or AC mass that I have told in the previous size. AC size exclusion, it will based on the seismic chromatography. It is seismic chromatography is based on the separation of a molecule based on this molecular weight. And AC sedimentation velocity, analytical ultracentrifuge. Then one is the thioplasmin T test, DLS, SDS phase, native phase, optical microscopy, NMR, F. These are the techniques for the characterization of the aggregates. Size exclusion chromatography. Size chromatography, as I explained, it is nothing but a chromatographic principle based on the chromatographic principle. It is based the separation is based on our hydrodynamic radius, nothing but the molecular size of the molecule. The molecule having the less molecular weight get retained into the pores of molecule. Molecule having the high molecular weight, they will unretain and they will elute the earlier. So if any aggregates are formed in your molecule, that peak will elute earlier, and it will show that if any peak is eluting earlier, it represents that there is a formation of aggregates or agglomerate in the molecule. With the help of ACC, also can conclude some fragments or transculated peptides. Transculated peptides or fragment is nothing but there is some deamidation, degradation of our peptides that you will get after your main analytes. If you are getting any peaks after your main analytes, it is representing that there is some transculation happening in your molecule. So this ACC in the ACC columns are available in the both. We can use for the HPLC as well as UPLC. So ACC column are silica based column or polymer based column. Both grades are coming in this. So generally soluble aggregates will get by using seismic chromatography. Generally, we are using the phosphate based buffer containing 100 to 500 millimolar sodium chloride at neutral pH. Sodium by using the sodium chloride, sometimes you will not get the proper peak shape, or you have the tailing or proper recovery of some fragmented peaks. So sodium chloride is replaced with sodium sulfate or sodium perchlorate. That will give you the better chromatographic peak shape. So high molecular weight or low molecular weight species can be detected by using this technique. High molecular weight species is nothing but aggregates, and low molecular weight species is nothing but your transculated or degraded peptides that you can get with this size exclusion chromatography. This is some of the data of the size exclusion chromatography of the proteins. So this is the main protein of the immunoglobulins. So if you are getting any peaks here like this in the red, suppose you are seeing fragment 1 or fragment 2. These are the transculated peptides. These are degraded and showing the transculation. If you are controlling this with the control, in the control that impurity at the lower rate, but if you are comparing with this real age sample, so you get this transculated peptide as, but there is no aggregates here. If you are comparing this second formulation, here the transculated peptides is not there, but you are getting the aggregates. Here same with this also. So from this we can conclude what is forming. Is a transculation or aggregation that we can determine by this technique. So generally during this development, we have to inject some standards. Molecular weight standard we have to inject based on this our molecular weights. So you will get some defined molecular weight standards and you will inject the samples. So based on this, you can determine where there is a formation of aggregates or transcription peptide. So this we will get in the EV, sec EV or sec mass. Generally, the sec mass is the robust technique that we are using for the characterization of the formation of aggregates.
तब दिया जाएगा ठीक है सर और उत्तराखंड से बिलोंग करते हैं ना सर तो सर
So this is the one application. Here what we have done, we have the 4 formulations with the different sugar moieties. These are the sucrose, threulose, and mannitol, and different process. That is the one is the lyophilized, one is the spray dyed. So we have drawn this data, and here we can get what percentage of the monomer is forming. So based on this, we can conclude that which polymer, which techniques, and which sugar moiety we have to use to optimize the formulation. We have to go with the lyophilization or spray dyed technique. This is the one of the technique for the aggregate dynamic light scattering, DLS. So DLS is one. It is based on the size distribution of our molecule, particle size distribution. Larger the particle size, there is the higher is the light scattering. So generally DLS is we are using in the range of 1 nanometer to 5 micrometer. If there is a formation of aggregates, that aggregates have the more mass or more molecular size. So that will give the more light scattering. So different. So this is not a quantitative technique. This is a qualitative. This generally we are using for the comparison of the different formulation. To compare the different formulation, we can use the DLS. To choose the different types of the excipients or effect of the excipients on the aggregation, we can select here. Also, we can study the effect of different pH and buffer concentration, as well as determine the thermal stability of our molecule. Or apart from the peptides, DLS is generally using for the suspension, emulsion product, nano suspension emulsions to properly optimize our particle size. So we can use this in the liposomal formulation. This is the some application of the DLS spectra. Here the 3 spectra are there, A, B, C. A is at the pH of 6.2, B is at the pH of the 4, and C is at the pH of the 8. If you are seeing this size distribution at a pH 6.2, your size is about 100 or near about 100 size. If you are going with the changing the pH 4, your size is about 1000. So what it's indicating? It is indicating that at the pH 4 there is a formation of the aggregates in your molecule. Same with the pH 8. pH 8 also we can feel something that there is a denaturation or some aggregates formation. So this from this data we can predict the optimal pH what is required for the formulation. This is the thioflavin T test. It is nothing but depending on the spectrophlorimetry principle. It is generally used to quantitate the fibril in your molecule. What were the fluorescence technique that we have seen? It is the intrinsic fluorescence technique. Here inbuilt, the molecule will absorb or fluorescence and give the spectra. This is the extrinsic fluorescence technique. Here, you have to add some dye in your molecule. Generally, we are using the thioplavin T. It is also known as the thioplavin T test. In this, because of the thioplavin T test, thioplavin T will bind to the aggregates and it will give the emission spectra at 472 nanometer. Or it generally bind to the aggregates, means it is the amyloid fibrils. So fibril will form at the later stage. First, you will get the aggregates. This is the general net2 structure. When there is a denaturation of a protein get misfolded. This will form the oligomer. Oligomer will lead to the generation of the prontofibrils or fibrils. This is the one mechanism. In this, when this oligomer have the secondary nucleation that we have explained, thermal, pH, or anything external phenomenon will happen that will lead to the formation of the fibril. When it is going the natural state, it will require some time to get the fibril formation. When there is any external factor with your oligomer, that leads to the accelerated condition of your fibril formation. So these fibril are detected by using this thioplamine T test. This is one of the important tests we have to use to prove this aggregation state formation. If by using this technique, if you have proven that there is a no aggregation, no fibril formation, then you can conclude that your product is okay. If there is a formation of your fibrils, so you have to work on the product development side. So this is the here. This is the spectra, fluorescence spectra of only thioplamine. You are not seeing any intense peak. This is the second spectra is only oligomer. Oligomer will not interact. Thioplamine T will not interact with oligomer. Will not show any intense peak. If there are these fibrils, so it will interact and it will show this. Sorry, it is monomer. It is not oligomer. Oligomer or fibril, it will interact the thioplamine T, and it will you will get some emissions, broad spectra, or absorbance here. So based on that, you can conclude that there is a formation of the fibril in your molecule. So thank you. This is from my side. If anyone have the questions, please. These are some regulatory requirement. This is nothing but a guideline. The guideline is for the peptide protein formulation that is there, that you can get online also.
Thank you, sir, for the wonderful session. Participants, if you have
Thank you,
any
sir.
doubts regarding the session, please feel free to comment in the chat box. Sir, we have one question here from Ms. Pooja Karpe.
The
In your
most
experience, what are the key factors to consider when selecting the most appropriate analytical technique for the specific research question or application?
Yeah, this question is regarding the research or industry you are asking.
Sir, good.
Hello. Yeah, it
Sir,
is.
you're audible.
Yeah, it is mainly depend on your molecule, which molecule you are chosen, or what data you require to characterize. That we have explained in some techniques. So generally, regulatory will not accept with only one analytical technique. So you have to use some orthogonal technique also, because these techniques have some pros and cons also. So you have to prove your data with minimum two orthogonal techniques. And techniques selection is depend on your molecular or which data you need for the submission.
One more question is there.
Yes.
In your experience, what are the key factors which are considered when selecting the most appropriate analytical technique for a specific research?
Mainly you can go with some spectroscopic technique, modern spectroscopic, because in spectroscopic you have the wide ranges of applications.
Shall we proceed for another question? Am I audible?
Yes.
Shall we proceed for another question? Can you discuss any innovative approach methodology you have developed to overcome challenges in sample?
Can you type in the chat box? Your voice is breaking.
Yes, sir. Question is in chat box.
Or analysis using advanced analytical technique.
Last one, that is innovative.
1 minute. Yeah. 1 minute. 1 minute. Can you discuss any need to approach or methodology? Or yeah, sure. So I will give example here. This NMR spectroscopy. So what were the we have the one formulation. In the formulation, we have the lactose and our peptides. So if you are getting the NMR signals, so the peptide have the less milligram, means some in the milligram range, and this excipient, whatever the lactose and mannitol we are using, that is about 200 mg. So in the 200 mg, we have the only 1 mg of the drug. So it's quite difficult to characterize that 1 mg drug with this 200 mg with this NMR spectroscopy. Because if you are considering the lactose, there are the number of the CS3 signals in this NMR region. So lactose will nullify your peptide signal and will not give the proper signal of this peptide. So here we have chosen the different approaches of how to remove this lactose from our API. So we have tried with precipitation, then dialysis membrane, then different types of pH gradient, pH isoelectrical. So in that we have got some data with dialysis membrane and with the proper pH, some selective pH. In the selective pH or by using the dialysis membrane, we have separated lactose from our main analyte. That is not in the 100%, somewhat reason, and we got some the peptide reason frequencies of this NMR spectra. So that will give us a tremendous idea of the chosen or selection of this technique to separate this lactose from our main analyte. This is the one example. If you are considering with the second one with also with the circular dichromism for EVCD. In for EVCD, suppose if you don't have some aromatic amino acids, in that case it is very difficult to get that tertiary structural determination. So we have tried with different concentrations or some extraneous chromophoric technique. So by using this technique, we got some conclusive data of the tertiary structural determination.
Get
So in if you are in the industry, so day to day, each day you have the different challenges.
Okay, thank you, sir. So we have another question. Have you ever encountered situation where unexpected results or artifacts arose during the characterization process, and how did you troubleshoot them?
Yeah, yes, we can get these circumstances. Generally, it will happen means two sides. One will be because of this, there is a problem with some formulations or problem with your analytical sites. If you are using any analytical instrument, if you are going with the basic instrument also, there is a troubleshooting is there. So before communicating any result or any conclusive data, so you should have the confidence on this data first. If you are in the disagreement or with any data, so you have to go thoroughly with that, and you can check the where the exactly problem will arises. So I have faced the one challenges with SU experiment. In SU we are doing with the third party. So they have failurized our product, one of the product that is optimized condition. So we have reviewed data and we go back to this laboratory and we have checked all the conditions. So there is some malfunctioning in their calibration of that instrument. So because of that, they have reported some tetramers or 100mers more times, and there is not matching with the reference product. So for this purpose, we have to look all this scenario. You have to first review your data before concluding or before proceeding to this any site.
Okay, sir. I hope this is clear to Ms. Renuka Thakne, ma'am. And sir, we have another question. What are some emerging trends or future directions in the field of advanced analytical characterization techniques? And how do you anticipate these developments will impact scientific research or industrial applications?
Yeah, now in the scenario, some hyphenated techniques are coming. Suppose you have to not depend on your one analytical technique. You have to pass that sample to the second analytical technique. Now, if you have heard about LCNMR, that is one instrument only available in the Nipper Mohali. There is no other way. In this LCNMR, you have to pass our LC analyte in the NMR signal. But that will give the data of the both the technique, mass spectrometry as well as the NMR spectrometry. Also, if you are going with this atomic absorption, AF4 and some microscopic techniques are advancing. Then one more is the time. Generally, when you are doing the same analysis, at the same you are doing at the room temperature, there is a denaturation or degradation of our molecule. At that time, you have to go for the time. So there is this technique. There is some advancement is there.
Thank you, sir. We have last question.
Yeah.
What piece of advice would you give to researchers or practitioners who are new to these techniques and facing obstacles in their implementation?
So first advice I will go to the researcher that before going to the any technique, you thoroughly read about the application of that technique, basic principle of that technique, and you have to learn somewhat troubleshoot of that technique, or you have to interpretation of that data. Whatever the data is generating from this technique, you have to interpret that data. And one more advice I want to give that you should not reliable on the single analytical technique. Always you should have the complementary or orthogonal technique to support your data.
I think we are clear with the question and answer session. Thank you so much, sir, for the wonderful session. Now I would like to invite Ms. Shubhangi Lingo to propose the vote of thanks.
Good afternoon to all the participants, esteemed guests, and our distinguished speaker, Mr. Dattatreya Vani, manager at PAR Formulation, Indo-Pharma. I, Ms. Shubhangi Lingot from B Pharma first year, feels immensely pleasured and honor to give vote of thanks on behalf of our management, principal, faculty, and all the organizing staff member of Yash Institute of Pharmacy, Chhatrapati Sambhajinagar. I extend my heartfelt gratitude to Mr. Dattatreya sir for his enlightening session on advanced analytical characterization techniques. His profound insight into this intrinsic subject have provided us with a deeper understanding of the cutting edge methodologies employed in pharmaceutical research and development. Mr. Wani's expertise has undoubtedly broadened our perspectives and equipped us with invaluable knowledge that will undoubtedly prove instrumental in our respective endeavors within the pharmaceutical industry. I would also like to express our gratitude to all the participants for their active engagement and insightful contribution throughout the session. Our eagerness to delve into the complexities of analytical characterization techniques reflects our commitment to excellence and continuous learning. It is throughout such a contributable efforts that we can collectively push the boundaries of innovation and drive positive changes within industry. Once again, thank you, Mr. Vani sir, for your enlightening presentation, and thank you to all the participants for your unwavering enthusiasm and dedication. We look forward to further opportunities to learn and grow together in pursuits of excellence. Respected sir, please accept a token of appreciation on behalf of the management and staff of Yash Institute of Pharmacy. We were delighted to have you here with us. Thank you, sir.
Thank you, sir. Thank you for the enlightening session.
Thank you. Thank you to the
And thank you
principal.
all the participants for joining. The queries regarding the feedback link will be provided tomorrow. And the next session for tomorrow will start at 10:30 a.m. sharp. Thank you.
Thank you.
Thank you, sir.
हाँ, जोशी sir बोले feedback link करना. बोलिए.
There is any link for the attendance of participants who joined and attended whole session of today.